Hello, my name is Anjali Vasavada, and I'm delighted to present our paper titled Methylation Markers for Anal Cancer Screening, a repeated cross-sectional analysis of people living with HIV as this week's IPVS Paper of the Week, published in the International Journal of Cancer. People living with HIV are at highest risk of anal cancer and will benefit from optimized screening for early disease detection. In our study, we compared host DNA methylation markers in high-grade squamous intraepithelial lesions versus samples negative for intraepithelial lesions or low-grade intraepithelial lesions in people living with HIV. We recruited people living with HIV identifying as male, age 18 years or older, undergoing high-resolution endoscopy in Seattle, Washington. Anal brush samples were collected for HPV detection, genotyping, and pyrosequencing methylation for host genes ASCL1, PAX1, FMN2, and ATP10A. We assessed associations between methylation and presence and extent of HSIL using generalized estimating equation logistic regression, adjusting for age, CD4 count, and HIV viral load. Marker panels using HPV DNA and methylations were also evaluated to predict prevalent HSIL. We found that increased methylation of ASCL1 and FMN2 were significantly associated with the presence of HSIL. These same markers were also positively associated with increasing HSIL extent. A panel combining methylation markers and HPV DNA showed the best balance of sensitivity and specificity for HSIL detection compared with methylation or HPV alone. From this study, we can conclude that host gene methylation testing shows promise for HSIL screening and triage.